So Alfred's hypothesis, two-hit hypothesis, is extremely crucial, as I said, to understand how we go from normal cells to cancer cells, or carcinogenesis, right? And I want you to understand that when he looked at this retinoblastoma and he saw, that the people who it had the inherited version of it had the two tumors in both eyes, and the people who were sporadic cancer only had it in one eye. Well, this can all be seen through this two-hit hypothesis, right? Because, as I said, they had this the people, the children who had the retinoblastoma as children, they had it very young, right, below the age of 5, and this can all be explained by this model right here. But if we, for example, look at lung cancer with cigarettes, suppose these both begin to smoke cigarettes at the age of 20. But why is that this guy has the malignant phenotype much earlier than this guy? And this is again due to the same phenomenon. This guy is heterozygous at birth, right? He has a mutant gene, which is this gene that has undergone a change or mutation in its DNA sequence. And this guy at birth is a wild type, right? He has the normal non-mutated version of the gene. But then this guy, when he smokes cancer, he will, let's say for 20 years, and he gets the cancer, boom. Now, he only has it in one here. And so he needs to be hit one more time for him to be homozygous and have the malignant phenotype. This guy only needs to be hit once. So he will be homozygous much earlier than this guy and get the malignant phenotype much earlier than this guy. Then the malignant phenotype will, of course, proliferate, and it will, of course, metastasize, and it would even kill him much earlier than this guy. Okay? So, unfortunately, or fortunately, however you want to look at it, cancer is due to both genetics and environment. Now let's talk a little about the cell cycle, right? So of course, cell enters the G1 phase, and then it goes through the cell cycle, as you guys know. But what are the drivers of the cell cycle? Well, the drivers of cell cycle are cyclins and CDKs. These are the drivers of the cell cycle. And I want you to understand that the regulation of cell growth is due to a bunch of different things. I'm not going to cover it all in this video, but I'm just going to give you a quick little overview. We have growth factors and receptors and transduction, specifically signal transduction molecules and things of that sort. And if you, for example, look at growth factors, so these are signaling molecules, typically proteins or peptides, that stimulate cell growth, right? And proliferation, and also even differentiation, right? So just understand that cells do not divide on their own. They need to be told when to divide. So we have some signaling molecules. We also call them growth factors, and these could be steroid hormones. These could be, for example, estrogen or something like that, which these growth factors bind to growth factor receptors and on the membrane, and then these receptors activate intracellular signaling pathways, which then often leads to a cascade of protein phosphorylation events, which then leads to a change in the gene expression. And the largest signal transducer is actually receptor tyrosine kinases. So this is actually also an enzyme. I'm not gonna talk about ligand binding and all these things right now in detail, because the time doesn't allow for it. But the reason why I've given you this overview is to understand a couple of things related to cancer. The first thing is tumor suppressor genes. So these are genes that suppress tumors, right? And these are very, very good, right? Don't misunderstand this. The way they work is, suppose you have something that enters the cell cycle and now proliferation just keeps on going. Well, we need to stop accelerating this process. And to do that, we have something that breaks down this acceleration of this process. And these are the tumor suppressor genes, right? So tumor suppressor genes in normal cells prevent cancer. And DNA changes that inactivate tumor suppressor genes can unfortunately lead to uncontrolled cell growth. And so tumor suppressor genes, as I said, are very good. But now suppose that they get inactivated. And normally it is through loss of function mutations. Or they can even get inactivated epigenetically. And both alleles normally should get inactivated for a phenotype to get observed. Then that makes a big trouble, because then you will keep on accelerating the proliferation process without apoptosis or even down-regulating the proliferation. But what about tumor suppressing genes and epigenetics, right? Well, to make it short here, remember I said that if hypermethylation is more than hypomethylation, if there is no balance, that could cause cancer. And this is because hypermethylation, well, then you're methylating a lot of things, right? Which suppresses genes, right? The RNA polymerase cannot read the DNA and transcribe it as normal. Well, that would cause problems and make genes silent. Now, suppose you have hypermethylation on tumor suppressor genes specifically, right? Well, what would that do? It will be very bad, right? Now you are prone to cancer, right? Because nothing can break down the cell cycle. And these tumor suppressor genes, they are your checkpoints. Their job is to say, do not divide if there is, for example, damage. Now, what about oncogenes? But to understand oncogenes, let's understand proto-oncogenes. So these are your normal everyday genes, which are responsible for basic cellular function in cells. However, if they get mutated, they can become an oncogene, right? And unlike tumor suppressor genes, oncogenes are dominant at the cellular level. So a single mutated allele is enough to contribute to cancer development, right? But what is oncogenes' job, right? Well, oncogenes' job is to accelerate the cell cycle, right? And so this, of course, will probably give you the phenotype of cancer. And what about oncogenes and epigenetics, right? So global hypomethylation of the genome can contribute to the development of cancer due to the fact that then you have more demethylation then methylation.